Welcome to the August 2023 Science in Action Seminar Series, where we focus on science, microbiome science at The Ohio State University that is utilizing the AMSL and the microbiome platform. I'd like to introduce Kristina Kigerl and Mohammed Mohssen for their talk on high-resolution genome result cut microbiome and viral dynamics after murine spinal cord injury. Thank you very much.
Thank you, and good afternoon. Well, I'm excited to be here this afternoon from the Department of Neuroscience to give a collaborative talk with my colleague Mohammed, who's been working with me and others on this project looking at high-resolution genome result gut microbiome and viral dynamics after mouse spinal cord injury. So I wanted to start by going through a little bit of background on spinal cord injury, introducing the problem that we're interested in. So currently in the United States, there are about 240 to 350,000 people that are living with spinal cord injury. And that's approximately 12,500 people sustained due to spinal cord injuries each year. And worldwide, there's about 250,000 people with spinal cord injury every year. The most common causes of spinal cord injury are motor vehicle accidents, followed by falls, violence, and sports-related accidents. Currently, 80% of all new spinal cord injuries are sustained by males. And the average age, although increasing, is now in the low 40s, as it was younger at 29. So most of these individuals are getting these traumatic spinal cord injuries at a fairly young age. They have a long time to live with their injury after. Oftentimes, when people think about a traumatic spinal cord injury, paralysis or the inability to move is the most common thought of problem. And although this is a significant factor that limits quality of life for people with spinal cord injury, there's really a lot more below the surface. So people with spinal cord injury deal with the loss of bladder and bowel control, loss of body temperature control, loss of sensation, and sexual and reproductive function. People with spinal cord injury have severely compromised immune function, so they're at high risk for developing infections really throughout their lifetime. Many digestive health issues. If you have a cervical level spinal cord injury, there are breathing difficulties. Many people develop cardiovascular disease, and this is often coexisting with earlier onset metabolic disease that develops, including things like increased adiposity and type 2 diabetes, neuropathic pain, frequent pressure sores, and muscle spasms. And there have been surveys done in individuals with spinal cord injury. And actually, when they talk about the factors that affect their day-to-day life the most significantly, it's often many of these comorbidities that actually impact their life more so than the paralysis itself. So in our lab, we really try to take a whole body systems approach to studying spinal cord injury. So we know that, yes, the spinal cord is the main connection between the brain and the musculoskeletal system, but it also is sending out spinal nerves and innervating really all the organs of the body. And the way that it does that is really through the autonomic nervous system. So to give a little overview of the autonomic nervous system, it's comprised of the sympathetic nervous system, which is the fight or flight response, and then the parasympathetic system, which is often called the rest and digest part of the nervous system. Most of the parasympathetic nervous system actually originates in the brainstem. And so if you have a spinal cord injury, the vast majority of the parasympathetic nervous system remains intact. However, the preganglionic sympathetic neurons live in this thoracic spinal cord. So T1 here stands for thoracic level 1 through thoracic level 12. And this sympathetic nervous system is really under the control of the brain under normal conditions. However, if you have a spinal cord injury in the cervical spinal cord or anywhere throughout the thoracic spinal cord, you're disrupting the brain's ability to communicate with the sympathetic nervous system below the level of injury. And this sympathetic nervous system innervates really every organ in the body. And so when we think about how spinal cord injury is causing some of these complications that are systemic, we think a lot about how disruption of the sympathetic nervous system could be contributing to that. So why is it important to study the microbiome after spinal cord injury? Well, we know that there is a bidirectional communication pathway between the microbiota in the gut and the brain and central nervous system. So under normal, healthy states, this remains in balance, and the brain can influence the gut and the microbiota living in the gut by secretion of neurotransmitters, neuromuscular control of peristalsis, through that sympathetic response or that fight or flight response, and also through the secretion of mucus. Conversely, the microbiota can also signal back to the brain, back to the central nervous system by production of metabolites such as short-chain fatty acids. It can signal through the vagus nerve by changes in barrier integrity. Our lab has focused a lot on immune signaling and systemic immune changes, and so we know the microbiota can change the systemic and the gut immune response, so things like cytokine production and secretory IgA. Again, the microbiota can produce neuropeptides and neurotransmitters, and these can all directly activate the vagus nerve. In addition, we know that disruption of the gut microbiota has been linked to numerous different diseases and different disease states. Many of these are things that are influenced by spinal cord injury. For example, we know that people with spinal cord injury have altered immune function, in particular in the lung as well as throughout the body. They also have gastrointestinal changes that occur. They have high levels of metabolic syndrome, insulin resistance, and obesity, as well as the development of liver pathology. So in particular, non-alcoholic fatty liver disease is something that forms in individuals with spinal cord injury, as well as high levels of depression and anxiety. So we wanted to first know whether spinal cord injury could be causing gut dysbiosis and whether it could influence any of these outcomes. So since the gut microbiota has been linked to many of these factors, we thought it could be a central way that it could be influencing the progression of some of these comorbidities that develop after spinal cord injury. When we first started in this field, we are not microbiologists, we're in the Department of Neuroscience, so we rather naively decided to take fecal samples from mice that had been given an experimental spinal cord injury, and then culture fecal samples on plates to see if we could detect any differences in the gut microbiota. And so when we did that, this is kind of what our plates looked like. And we did see that after spinal cord injury, we saw a change in the number and types of colonies that we were able to culture on these plates. And we recognized that this was a very crude and limited way of looking at the gut microbiota because the vast majority of the microbes in the gut are not cultured in this way. So we then decided to do 16S sequencing to look at the composition of the bacteria in the gut. And hopefully what you can appreciate from this heat map is that most of the spinal cord injury samples shown here in red cluster on the left side of this heat map. Then most of our naive samples or samples from animals that had a surgical controlled surgery in blue here are clustered together on the right side of this heat map. And here we're just looking at the major bacterial orders that are found in the gut microbiota. And so what we noticed over time after injury and compared to our surgical control is that the percent and abundance of the bacteroideals decrease over time while the clostridials increase. So we knew from this data that gut dysbiosis was occurring after spinal cord injury, at least through 28 days post-injury, but we didn't have any readout to determine whether this dysbiosis influenced outcome from our injury. And so we decided to approach this question using a couple different techniques. First, we wondered, can we make the dysbiosis worse? What if we take animals that have a spinal cord injury and we give them antibiotics? Antibiotic use is extremely common after spinal cord injury because the vast majority of people that have a spinal cord injury have surgery acutely to stabilize the spine. And so they're given prophylactic, pre-surgical and post-surgical antibiotics. In addition, the rate of infection in people with spinal cord injury, especially pneumonia and bladder infections, is very high. So they're frequently on antibiotics throughout their life. So we thought we could try to disrupt the gut microbiota, giving antibiotics in our animal model, and then see whether that affected outcome. And so we actually started the antibiotics a week prior to injury. And the purpose of this was to really be able to test sort of a proof of concept of whether having a disrupted gut microbiota at the time of injury would affect outcome. And then we collected fecal samples from these mice and did some behavioral analyses through 35 days post-injury. What we found is this scale here that says VMS score is actually a scale for locomotor recovery. It looks at hind limb motion in animals after spinal cord injury. And so a score of 9 would be normal locomotion, and a score of 0 would be complete hind limb paralysis. And what we found is that in our animals that had induced dysbiosis with the antibiotics, so those are these light squares, they had reduced locomotor recovery compared to our spinal cord injured animals alone. So this suggested that the dysbiosis was making the spinal cord injury worse. When we looked at their spinal cord, so this is a cross section of a spinal cord after injury that is stained for axons and myelin in blue and brown. And what we saw and quantified here is that the animals that had induced dysbiosis with the antibiotics actually had larger lesions and then less of this spare tissue. And it's spare tissue is important because this is the tissue that typically mediates recovery after injury. We also stained the spinal cord for different inflammatory and immune cell markers and found that there was greater presence of macrophages, B cells, and trend towards increased T cells that had responded in the spinal cord to this injury. So we were able to show that when you get a spinal cord injury, you can cause some gut dysbiosis, and that this gut dysbiosis then can impair recovery if it's to a certain degree. So that led us to hypothesize, well, what if we try to deliver probiotics? Can we reverse this gut dysbiosis by delivering probiotics? And so we chose BSL number 3 as our probiotic to start with. And the reason that we chose this really is because it contained 8 different strains of probiotic bacteria. When we began looking in the literature and trying to decide which probiotic would be best, it was difficult to determine based on what was out there because nobody had looked in spinal cord injury at probiotics. So we picked this really because we felt like we were kind of throwing a lot at it at the same time because there were these 8 different species. So what we did is we again used our mouse model of spinal cord injury, and then we started feeding them this probiotic every day, starting on the day of injury. And really, much to our surprise, because we didn't think the probiotics would have an effect on locomotor recovery, we did see that when we treated every day with this high dose probiotic, we were able to slightly improve locomotor recovery after spinal cord injury. And again, we saw that the lesion volume was smaller and that the probiotics increased the amount of that spare tissue that we found to be important for mediating recovery. When we looked at the fecal samples from these animals that had been given probiotics, we were able to see that the 2 bacterial families that were present in the probiotic were increased, in particular at 7 days. So this was a 16S thing. So at 7 days, you could detect an increased abundance in the bifidobacterium and lactobacillus. And this was still elevated compared to our control water-treated animals over time, but not as high as 7 days. Interestingly enough, when we looked at the family level, it was really the bifidobacterium in this pink triangle here and the streptococcus strain that seemed to be increased the lactobacillus, is this bright green sliver here. It remained unchanged, even though half of the probiotic we were feeding was lactobacillus. But we don't really think that giving people commercial probiotics after spinal cord injury is going to greatly improve their recovery from injury. But this was really more of a sort of proof of concept study that we can either make the microbiota, we can modulate the microbiota and have an effect on some of our outcomes. But we really became interested in trying to apply more robust techniques to studying the microbiome. And this is where our we were very fortunate to start collaborating with the Sullivan Lab and Matt Sullivan's group on helping us to do some whole genome shotgun sequencing and metagenomics. So we decided that we wanted to sort of expand on our initial experimental design. So in our initial design where we did the 16S sequencing, we only used a single injury level, which was a T10, and we only kept our animals out for 28 days post spinal cord injury. And when we had shared that data after it was published, a lot of people asked us, well, does injury level affect this? So you gave an injury, you know, sort of at the bottom part of the spinal cord. What if you had a cervical injury or a higher level injury? People also asked, you know, how long does cystitis last? Does it persist beyond the first couple of weeks, or is it just a temporary thing that will go away? So we decided to do 3 groups. Our laminectomy group is a surgical control. So these animals receive everything the same as the other animals that have a spinal cord injury. And we remove the dorsal surface of the vertebral lamina that is covering the spinal cord, but the spinal cord itself is not injured. Then we gave a T4, so thoracic level 4 spinal cord injury, or a T10. And we chose these 2 levels based on what is known about the sympathetic nervous system. So in a mouse at T4, if you give a spinal cord injury, you're really cutting off the brain control. So pretty much all of the sympathetic preganglionic neurons that live in the spinal cord. So you're disrupting its brain from signaling to any of the sympathetic nervous system. Whereas if you get a spinal cord injury at T10, you're really only disrupting communication to this bottom part that tends to innervate the colon more specifically. We never use antibiotics in these animals because we really want to be able to assess whether the changes we're seeing are due to the spinal cord injury and not due to an antibiotic effect. We single house them so that their microbes are not shared among cage mates. And we don't change their diet at all throughout the study so that dietary intake is controlled. So we kind of also wanted to, in addition to looking at these different injury levels and looking across time, we decided to keep these animals for 6 months post spinal cord injury and collect those samples through 6 months. And we also wanted to do a sex comparison between males and females. Our previous study had been done only in females, and female animals are commonly used in spinal cord injury research because they bladder care. So just like people who have spinal cord injury, animals that get spinal cord injured also lose the ability to control their bladder. And so the type of post surgical care that is required to maintain bladder function after spinal cord injury is easier in female mice than in male mice. However, the majority of people with spinal cord injury are in fact males. And so it was important to do this sex comparison as well. And so we collected hundreds of samples for metagenomics, and then we kind of realized that this was going to be very expensive and very time consuming, very difficult to do the analysis. And so before moving on with all of our samples, we decided to analyze just a subset of our samples. So only 15, 5 per group, so 5 laminectomies, T10 or T4s. We chose 21 days because that was a time point in our previous 16S study that we had already seen the development of gut dysbiosis, and we started with female samples again because we wanted to see if we could sort of replicate with the metagenomic data what we had seen previously with our 16S data set. And so this paper was published a couple years ago, and I'm not going to go through most of the data, But I just wanted to show briefly that when we look at the microbial component of the gut, in those 15 samples, you can again see that our spinal cord injured samples cluster very distinctly from our laminectomy samples. And it seems like this dysbiosis is actually enhanced in the higher level of the T4 spinal cord injury. So this is Gray-Curtis dissimilarity scores between the spinal cord injured groups and the laminectomy controls. And so this dissimilarity is greater in the T4 group compared to the T10. And we do see some changes in several phyla at this level. When we look at the species level and in this injury time point, just again, this little snapshot at 21 days, what we found were that there were spinal cord injury dependent and spinal cord injury level dependent changes in the bacterial species. So cluster 1 and cluster 3 are really bacterial species that are changing due to spinal cord injury, but not dependent on the level of the spinal cord injury. So this cluster 1 are species that are decreased in both the T10 injury level and the T4 injury level, whereas cluster 3 are species that were increased in both. However, cluster 2 showed a level dependent change. So these are bacterial species that were only increased in this higher level spinal cord injury. So overall, it seems like this T4 injury is just causing greater dysbiosis. And then we looked at some of the potential species that were decreased and increased after injury. Another big benefit of this approach is the ability to look at the functional capability of the microbiome and start to look at the metabolism. And so since we had done whole genome shotgun sequencing and we had metagenome assembled genomes from our sequencing, we were then able to look at the gene pathways in those species and see what was changed. So this is only a sample of those changes, but again, when we do a PCOA analysis of the genes, we see that the composition of the genes in our microbiome is significantly altered after spinal cord injury compared to our laminectomies. And then when we looked at some functions that we felt like could be important, especially related to central nervous system function after injury, we saw that in both T4 and T10, these functions were decreased. And so these include things like tryptophan biosynthesis. So tryptophan is the serotonin precursor, vitamin B6 metabolism, and folate biosynthesis. Lactoseven is an anti-inflammatory peptidase that was reduced after spinal cord injury. The phosphotransferase system is involved in carbohydrate utilization. Glycoside hydrolysis can be involved in short-chain fatty acid production. And then there are many other changes that are not shown on this slide that are published in this manuscript, showing that not only is it changing the abundance of the community members, but also the functional potential of that microbiome. One benefit that we were really excited about after this study was the fact that because we had these metagenome assembled genomes, we were then able to map the functions that we were interested in back to their hosts. So for example, this first column here is Lactobacillus johnsoni, and this was a bacterial species that was really highly prevalent in our laminectomy animals, but was significantly decreased in both of our spinal cord injury groups at both T10 and T4. And so then we could look at these specific gene families and see what functions that MAG contain. And same with the MAGs that were then increased after spinal cord injury. So because of the data and approach from that paper, we then decided that this was the way that we wanted to move forward with all of the rest of our samples. And so this is where Mohammed came into the story. And I'm going to let him take over from here because he has really been the driving force on the analysis of all of these hundreds of samples.
So the first thing I want to talk about is the difference between 16S
and metagenomics. So what we can call systems for Amplicon sequencing metagenomics. And the reason is we want to emphasize why we're doing this. So when we talk about Amplicon sequencing, we just talk about sequencing these highly conserved 16S genes that are in all bacteria. And then these genes, these reads, you map them to a reference database and then get an idea about the taxonomy of these. The problem with that is that because these are highly conserved, as quickly as the other functional genes. You do miss a lot of the functional potential. We're only limited to what you see in the database. You also get a lot low level taxonomy, so you get the order of order or family, and you miss other organs like viruses. So the reason why people do shotgun sequencing or metagenomic sequencing is that they sequence the whole genome of everything in the sequence. So you get these short reads that you then either map again to reference genomes, and now you can get an idea about the functional potential. You can get an idea about what are these genes are functioning in your system. But you also miss a lot of what you not of your organs, and it's not contained in reference genomes. If they've all more quickly or if the genome is not complete for instance. So the other thing you can do is what we call genome result metagenomics, is that you can assembly these reads into context. These are overlapping sequences. The sequences of DNA, and then you bend these sequences based on coverage, nucleotide composition, and so on, into what we call traditional assembled genes or MANs. These MANs you can now map these group reads with these MANs, and you get a much better idea about who's there, what they're doing, and what the taxonomy is. So this is what we did. We got these short reads, we see them and track them, assemble them into something called co-assembly. We assemble 5 mouse, and we bend them into different mags. We replicate, and then we have a final list of mags that we look at. Of course, back then we didn't have cancel, so we did that ourselves with a safe, of course, 6 month order to be we just had someone do that. And we had around 80% of our reads mapping to these mags, so which means we covered at least a portion of our sample. Now we wanted to ask, okay, how many viruses and bacterial genomes are we recovering? Are we seeing the same patterns of viral change after SCI, and how did they change, and what are these phenotypes? We wanted to get at these species, or that's what we want to isolate and maybe have clinical so we did recover a lot more mags, around double what we had from retail, which was a lot more than had previously. And we did recover around 26 times what we had from retail, 26 times viruses. And most of them are high quality genomes. Want to talk about that. So if you look at, as Kristina was saying, most of the studies, if not all studies, were done on female samples, female lines, while most of the patients are male. Uh, so we wanted to see if the microbiome is sex specific, and it turns out that it is. Even pre-3, there is clear separation between microbes in male versus female mice. So that's the first thing. I wanted to see if there is a level dependent or just an SI dependent injury effect. So we were seeing in the account because there is male and female. And to answer this question, okay, how much of this change is persistent? So if we plot greater consistencies relative to 0 days, which is like the injury, you see that starting 7 days and up to 63 days, there is a clear difference between spinal cord injury, injury microbiome, and minor. Suggests that the changes we're seeing are actually consistent. So next, we do the pairwise comparisons, just looking at which of these taxa are differentially funny in treatments, treated with the environment before. And if you look at phylum level, you're seeing the same things we're seeing in the literature. The firmicutes are higher after injury and bacteria are lower. But if you look at the species level, we get a lot bigger, bigger list of taxa species that are changing. Some of them are high abundant, some of them are medium, some of them are low, and we really don't know what is changing or what is driving these changes after that step. So we also know that from the literature, from other experiments, that bacteria do change behavior if they are cohabited with different bacteria. So there is a well-known example of the Victoria's Amplicon, where the polysaccharide neutralization is different. The transcripts are different when you get cohabited with different bacteria. So we know that we're missing a lot of these interspecies interactions just by looking at each mag changing alone, and that these interactions could be driving this carbon utilization and immune system function that we hope to understand. We also know that if we want to give probiotics to people, we know that if we give, you have to really understand what microbe is perturbing all the other microbes. So we resorted to one known systems biology approach. It's called WTNA, where you basically cluster your coverage profile for all of these mags. So you get like these two mags have like similar cluster profile than these, and then you get modules of changing taxa. So instead of just looking at one taxa at a time and comparing it to your system output or system management, like the injury, you now can look at the modules and correlate them to the system. And we did that per time point, and you can see at each time point there are certain modules that are changing. These modules, if you compare them to BMS score, Chrissy explained just how well these mice can walk. You can tell something about their importance in each of these methods. This is also led by So now we're able to see at each time point what are these bacteria in each module. how they are changing, how they are interacting with each other, and so on. And we're seeing most of these bacteria are some that we saw before, some of them are new, but we're now making sure that these are the ones that are driving change, not because we've seen others in these business and good health. Now we're able to narrow down our focus and try to isolate. So now we're up to 19 mags driving these changes on the recent one. And there are some that change with like increase with injury or some that decrease with injury. And by the way, not every one of them has to be like the most abundant or very abundant bacteria in the gut. Some of them are actually low abundant on the level of them, but they seem to be important. So the next steps would be looking at these mags metabolically. We can functional enrichment analysis. We can check which of these genomes, which of the genes are conserved versus which of them are not. And we can see these interspecies interactions in the population genomics and understand the roles of viruses. And so also help us with AMGs that have not included this results, but that is when. So yeah, these are acknowledge that, accomplish that, solve that. First time in CM, just here with him today. Worked together for 2 or 3 years now. Day and Ahmed.
Mohammed, that it's really exciting to see that you've made the progress in the WGCNA analyses. I know this was really difficult, both with the strain level variation that you had to tease through, and then the longitudinal problems of applying the existing WGCNA workflow. Are there parts of this analysis that you feel like still need significant work, or do you feel like you've really conquered the kind of time resolved longitudinal application of WGCNA?
For this, I would say that